Hi, everybody. Mrs. Murphy here. Hope you guys had a great break. I'm just going to go over approximately 10, 11 of your progress checks. So I'm going to go ahead and get started with question number 1. And so I'm just going to go ahead and look at this question. It says scientists conducted a transformation experiment using E. coli bacteria and the P. true plasmid. Samples of the P. true plasmid, lane A. And that's right there. And the chromosomal DNA from 2 different E. coli strains that scientists attempted to transform, lane B and C, were compared using gel electrophoresis. The results are shown in figure 1. The question is, which of the following statements best explains the experimental results observed in figure 1? All right, so B and C, all right, that's the chromosomal DNA from 2 different E. coli strains. All right, so that's your B and your C. This is simply, A is simply the plasmid. So again, which of the following statements best explains the experimental result observed in figure 1? All right, so the answer is C. E. coli and lane C have successfully transformed and contain additional genetic information. All right, and the reason is because this one here, and I will scroll up so you can see, sorry for my lines here, but if you have the transformation, you're going to have the chromosomal DNA and the plasmid. So you can see that lane C here, I want to do a different. Lane C here is going to have the chromosomal DNA and it's going to have the plasmid. So if I go back up, okay, it says chromosomal DNA from 2 different E. coli strains. So it has both. And then when I look at the explanation, and let me get this out of your way now. It says a successful transformation would contain a band of DNA from the P. true plasmid and a band from the DNA from the E. coli chromosome. Lane C has both bands and is evidence of a successful bacterial transformation. So showing that it has the plasmid and it has its original DNA. All right, so D, which E. coli have been transformed can't be determined. That's not true because you can see that. B. E. coli and lane B have been successfully transformed and contain additional genetic information. No, because you can't tell that the plasmid's there. So showing the plasmid, remember the plasmid, the gene would have to be cut and then it'd have to be put in. And A, E. coli in both lanes B and C have successfully transformed and contained additional genetic information. So it's not B and C. There are 2 strains of E. coli, so only C shows having the plasmid. All right, moving on. Which of the following best explains how the pattern of DNA arrangement in chromosomes could be used in most cases to determine if an organism was a prokaryote or a eukaryote? All right, so they're talking in all of these choices about prokaryotic and eukaryotic DNA. And so basically you should be thinking in your head, prokaryotic DNA is circular and it's a single chromosome. DNA is linear. All right, and there's many, we know that we have 23 pairs of chromosomes. So this one is just checking to see if you know your difference between prokaryotic and eukaryotic DNA. So if we look at A, single circle chromosome, that is correct. Multiple circular chromosomes, that is not right. B, prokaryotic DNA, multiple chromosomes, that's wrong. Eukaryotic DNA, single chromosomes, that's wrong. Prokaryotic DNA for C, single linear chromosome, that is wrong because it's linear. Multiple linear chromosomes for eukaryotic, that is correct. And if we look at prokaryotic DNA, for D, it's a single circular chromosome, which that is correct. Eukaryotic DNA, multiple linear chromosomes, that is correct. All right, so going on to question 3. Lynch syndrome is an inherited condition associated with an increased risk for colon cancer, as well as certain other cancers. Mutations in one of several genes involved in DNA repair during DNA replication have been associated with Lynch syndrome. DNA sequencing was performed for an individual. The results indicated that the individual carries one of the dominant alleles that has been associated with Lynch syndrome. So remember, Lynch syndrome is associated with an increased risk for colon cancer, as you know, right here. So right here is an inherited condition associated with an increased risk for colon cancer as well as certain other cancers. All right, so if someone has that dominant allele that's been associated with Lynch syndrome, it says which of the following best explains how the results should be interpreted. So let's look at the choices here. So it says A, the individuals does not have increased risk of developing cancer because one dominant allele is insufficient to cause the disease. So it already, like it tells you that the results indicate the individual carries one of the dominant allele and that has been associated with Lynch syndrome. So that's going to be wrong. All right, let's look at C because it already tells us B is right. Because the person's DNA has the mutation, other family members must have the cancer. That's wrong. It's going to tell you up in the reading that mutations in several genes, say during replication, so it's a replication error. That doesn't mean that it got passed on from parent to offspring. So that is incorrect. Results cannot be interpreted until testing determines if additional mutated alleles are present. That is not right. Remember, it said it's associated with other cancers. So B, the individual has an increased risk of developing colon cancer. It tells you that. It tells you alleles associated with it. So that is the correct answer. All right, number 4. Number 4. An evolutionary biologist hypothesizes that two morphologically, okay, that means shape, similar plant species are not closely related. To test the hypothesis, the biologist collects DNA samples from each of the two plant species, then uses restriction enzymes to cut the DNA samples into fragments, which are then subjected to gel electrophoresis. All right, so you get these species, restriction enzyme cuts them, they're running the gel. The results are shown here. All right, so there's A and that's B. The question is, given the results shown in figure 1, which of the following correctly describes the relationship between the two species? All right, so this is simply you're looking at the data and what you know about gel electrophoresis, right? So you have to remember in your mind, oh, gel electrophoresis separates DNA by their size. The small fragments move farther than the large ones, right? And so remember, DNA is negative, so it's going to go to the positive pole. So let's keep those things in perspective and in the back of our minds when we read these choices. So let's look at these. Species B is the ancestor of species A because it has fewer bands. Whoops. When we look at this, there's nothing that you can tell that one is the ancestor over the other, and fewer bands is not going to indicate that. All right, so that one is going to be incorrect. Species A is more complex than species B because it has more bands. No, that just means the restriction enzyme goes and cuts it at different sequences. So that is incorrect as well. Letter D, species A has more short fragments than species B does. All right, so the question is, which of the following correctly describes the relationship between the two species? I don't know if that is a relationship. It has more short fragments than species B. Let's see what it says here. So if I go and look at this, species A has more short fragments. Well, actually, which one has more short fragments? It's going to be B, right? So B has more short fragments because look at how many small ones there are here. The shorter ones travel farther. So that is actually incorrect. But let's look at what C says now. Species B has more short fragments of DNA than species A. All right, so before I was like, I don't know about this relationship, but like I know A and B are wrong, and now you're, looking. The only skill you have to know here is that gel electrophoresis is separating on size. So what do you have to, pick here? Yeah, these fragments are smaller. It means that the restriction enzyme cuts species B DNA into smaller fragments than A. So that, of all the choices, is simply the only relationship that is seen here. All right, so that is the correct answer. And these should be released to you so you guys can see these. I did do that in the assignment. All right, moving on to number 5. Oh, a DNA replication problem. All right, it says labeled nucleotides were supplied to a cell culture before the cells began DNA replication. A simplified representation of the process for a short segment of DNA is shown in figure 1. Labeled DNA bases are indicated with an asterisk. All right, so where are those at? Those you can see here. Spotlight does not work. Oh, right here. These asterisks, I don't know if you can see my mouse, but they're right here. You can draw and circle them. All right, there's your labeled DNA bases. All right, so showing you the new ones that are coming in. All right, so what's the question? Which of the following best helps explain how the process represented in figure 1 produces DNA molecules that are hybrids of the original and the newly synthesized strands? All right, so this question is asking to see if you understand the semi-conservative model of DNA replication. So remember in DNA replication, when a new DNA molecule is made, you get one template strand and one new strand. And remember, that's the semi-conservative one. So let's see what is said here. So each template strand is broken down into nucleotides, which are then used to synthesize both strands of new DNA. All right, that does not happen. The template strand doesn't get broken into pieces of nucleotides. Remember, your nucleotide is a sugar, a phosphate, and one nitrogenous base. And then those aren't used to make a new one. So that's wrong. B, each template strand is broken into multiple fragments, which are randomly assembled into two different DNA molecules. That is incorrect. C, each newly synthesized strand is associated with another newly synthesized strand to form a new DNA molecule. No, that would mean that there's no original strand, there's no template and a new. That means that there's just two new. So that is incorrect. And the answer is going to be D. Each newly synthesized strand remains associated with its templated strand to form two copies of the original DNA molecule. So that is going to be the correct answer. All right, moving on to question 6. Cyclohexamide is a eukaryotic protein synthesis inhibitor. It is used in biomedical research to inhibit protein synthesis in eukaryotic cells studied in vitro, so like in the lab. Its effects are rapidly reversed by simply removing it from the culture medium. In a translation experiment using fungus culture, radio-labeled amino acids were added to the culture, allowing the researchers to measure the growth of a single polypeptide chain. By measuring counts per minute, as the chain grew, the CPM increased. After a certain amount of time, CHX, remember CHX up here, okay, is a protein synthesis inhibitor, was added to the mixture and the experiment continued. After an additional amount of time, CHX was removed from the culture medium. So again, it's an inhibitor, and it says it's used in research to inhibit protein synthesis. Its effects are rapidly reversed by simply removing it from the culture medium. All right, so let's look at what this question is. It says right here, which of the following graphs best predicts the data collected during the experiment? All right, which of the following graphs best predicts the data collection during the experiment? Again, amino acids are added. All right, they're growing polypeptides. Then again, it says they added the CHX. Then it says they removed it. So let's take a look at these graphs. So if I could ever get my computer straight. So let's see. When I just look at this one, it says an inhibitor is added right here. Okay, and this is an inhibitor remove. If it's an inhibitor, why would it continue to go up? So I'm going to say no way to that one. So here's an inhibitor added. Okay, and it looks like it goes down. And then inhibitor removed, it goes back up. All right, that one for me is a maybe right now. Okay, so I'm not sure about it decreasing it, but it does, you know, it doesn't continue to go up and it does go back up when it's removed. This one, an inhibitor added, inhibitor removed, that doesn't make sense. Right now, A and C are out. All right, so, okay, inhibitor added, looks like nothing's happening, it's steady. So remember, this is the counts per minute. All right, so then an inhibitor removed, it looks like it goes up. Well, let's go back up to the reading again, because you know you're always between two when you're doing these questions. So let's see. Says amino acids were added to the culture, allowing research to measure the growth of a single polypeptide by measuring counts per minute as the chain grew. After period of time, CH was added and then continued after additional amount of time. It just says it's an inhibitor, meaning it stops it. It doesn't say it decreases. So based on that, so I went back up to the first paragraph too, you guys. So I think that based on what that says, just inhibited protein synthesis, and then it's rapidly reversed. And you know, they're just measuring the counts per minute. So it doesn't say it decreases or like breaks it down into polypeptides or something like that. So that's why I say B is wrong, and then we're going to go to D, which is the answer. All right. I don't know where my plasmid's coming. I'm doing the undo button. Maybe if I do redo. It's really weird. I'm doing redo. Come on. So I'm going to go back now. We're going to go to number 7. All right. Number 7 says retroviruses such as HIV and hepatitis B virus use RNAs or genetic material rather than DNA. In addition, they contain molecules of reverse transcriptase, an enzyme that uses an RNA template to synthesize complementary DNA. Which of the following best predicts what will happen when a normal cell is exposed to a retrovirus? All right, so you got to be thinking, if you know what retroviruses do, they go in as DNA, they reverse transcribe their RNA into the DNA, into the host genome, and then it gets transcribed and translated. All right, so let's look at A. The reverse transcriptase will cut the host DNA into fragments, destroying the host cell. All right, if you know what a retrovirus does, you know that that does not happen. If you know generally how this happens, you would be able to rule that out. The reverse transcriptase will insert viral RNA into the host genome so it can be transcribed and translated. Remember, it violates the central dogma because it's retrovirus, so that's kind of close, but it's not going to be that because it goes from RNA to DNA. C, the reverse transcriptase will produce DNA from the viral RNA, which can be incorporated in the host genomes and then transcribed and translated. So that's correct. Well, let's look at D. The reverse transcriptase will force the host ribosomes to translate the viral RNA prior to polypeptide assembly. That is incorrect. All right, that hopefully was an easy one. All right, let's go on to number 8. Exposure to ultraviolet UV radiation is the leading cause of skin cancer in humans. Figure 1 shows a model of how UV exposure damages DNA. All right, so you have your before on the left and after on the right. And one thing that I noticed about the after is this big bulge right here. All right, and that's like a dimer. All right, so let's look at the question. Which of the following statements best explains what is shown in figure 1? UV exposure triggers DNA replication, which results in rapid cell proliferation. No, that does not occur. Naturally occurring dimers are removed by the UV photons, causing misshapen DNA, which results in replication errors. No, so it does not, they don't get removed by photons of light. So that is just incorrect. C, the hydrogen bonds between the base pairs absorb the UV photons, separating the two DNA strands, which results in rapid DNA replication. That is incorrect as well. Okay, those hydrogen bonds are not absorbing UV photons. UV photons cause dimers to form, leading to misshapen DNA, which results in replication and transcription errors. All right, so that is the correct answer. And remember that we do have mismatch repair enzymes to repair and fix this. But there are conditions where people have where they have mutations in their mismatch repair enzymes and their nuclease enzymes. Okay, so let's go on to number 9. Oh, we got an operator. All right, we got an operon. And one thing you should be thinking about immediately is prokaryotes. All right, the TRP operon in E. coli is an example of a repressible operon that consists of genes coding for enzymes used to synthesize tryptophan. When tryptophan levels are high, the operon is turned off and these genes are not transcribed. However, it is also known that tryptophan does not bind directly to the operator DNA sequence. The regulatory gene called TRPR has also been discovered, although it is not part of the TRP operon. The proposed model of how the tryptophan acts as a co-repressor is shown in figure 1. All right, so if you're looking at this, remember here's the regulatory gene, that TRPR. Okay, it's not part of the operon because this tells you right here, this is all of the operon. Should have cut in about right there, sorry. And remember, this gene makes the inactive TRP repressor. All right, so this is inactive, this is active. Remember, when it's active, it can go ahead and it can bind in the operator. And when it does that, then that shuts the pathway down. Because remember, this one is always on. This is a repressible operon. It can be making tryptophan, but then if tryptophan is present, okay, it binds. Okay, it binds. Remember, if it was tryptophan, it could be like something that would then bind. Once it would bind here, then it would activate that, right? Activate the repressor, then it could bind. All right, well, let's see what the question asks. Which of the following evidence best supports a claim that tryptophan functions as a co-repressor? All right, so we have learned this. So let's see. Well, the question is, which of the following evidence best supports the claim that tryptophan functions as a co-repressor? All right, normal expression of TRPR causes the TRP operon to be transcribed regardless of tryptophan levels. All right, so that's not true. Normal expression of TRPR. Okay, so you can even see normal expression, you get an inactive one. All right, and you don't really have evidence of that from that picture of, regardless of tryptophan levels, right? Because it's not showing you the tryptophan levels. It's not showing you whether there's some there or not. When the operator sequence is mutated, the TRP operon is not transcribed. All right, so there's no evidence of that either. Okay, so the operator sequence. So, that's the operators up here. The TRPR gene codes for a repressor protein that has a DNA binding domain. It does not show that. The answer is D. When TRPR is mutated, the TRP operon is transcribed regardless of tryptophan levels. All right, so let's see what the answer says here. As tryptophan accumulates in the cell, it inhibits its own production by activating the TRP repressor protein. All right, which then binds the operator blocks. Okay, so this is what I went over earlier. If the TRPR gene is mutated, then tryptophan cannot bind to the allosteric binding site to activate TRP repressor protein. Thus, no matter how high the tryptophan levels get, the TRP operon will not be repressed. All right, so when the TRPR is mutated, so again, the TRPR is right here. The TRP operon is transcribed regardless of tryptophan levels. So basically, you mutate this, you're not going to get your repressor. So it doesn't, regardless if there's tryptophan present or not, it's not going to play a role in whether that repressor is in the operator or not. So that's why that's the answer. All right, question number 10. Eukaryotes transcribe RNA from DNA that contain introns and exons. Alternative splicing is one of the post-transcriptional modifications that can create distinct mature messenger RNA molecules that lead to production of different proteins from the same gene. Figure 1 shows a gene and the RNA produced after transcription and after alternative splicing. Okay. So let's see, you got your exons 1 through 5, transcription. You got your pre-messenger RNA exons 1 through 5, and then after alternative splicing, you can see you're going to get different enzymes because different sequences are considered introns and intervene, and others are considered exons that express. So you're going to get A, B, and C. So if it is to, let's see. All right, let's see. A cell needs to metabolize the substrate illustrated in figure 1 for vital cellular function. Which of the following best explains the long-term effect of the cell splicing that yields only enzyme C? So let's take a look at enzyme C. Enzyme C looks like this. Remember how an enzyme works? It's an enzyme and a substrate, and to me, that does not look like those are going to fit together. It looks like that substrate fits into A. All right, so to me, it's going to be a problem between the substrate and C. So if the cell splices and only produces C, it's not going to be able to metabolize, okay, without enzyme A. All right, so the answer to this question is letter A. Let's see what the other choices say. B says the cell will remain healthy because all 3 of the above enzymes can metabolize the substrate. All right, you guys should be able to see that they don't fit. That's not going to work. Let's look at C. The cell will remain healthy because enzyme C, messenger RNA, will undergo alternative splicing again until transformed into enzyme A, messenger RNA. All right, you've learned the processes that does not happen. It can't just turn itself into another one. D, the cell will remain healthy because enzyme substrate interactions are non-specific, and enzyme C will eventually metabolize the substrate. All right, enzymes and substrates are specific, so that is incorrect. Okay, question 11. A simplified model of DNA replication fork is represented in figure 1. The protein labeled enzyme 1 carries out a specific role in DNA replication process. In the DNA replication process, sorry. Which of the following statements best explains the role of enzyme 1? All right, enzyme 1, when you look at this, okay, you should be thinking that's topoisomerase. It's upstream of the replication fork, so let's see what the choices are. Enzyme 1 is DNA ligase. Oh my goodness, if you're picking that, I am upset. All right, so that one is, that is not DNA ligase. DNA ligase, you should be thinking in your head that seals fragments of DNA together. Enzyme 1 is DNA primase. No, that catalyzes the synthesis of the primers. Oh goodness, no, that is incorrect. So C, enzyme 1 is DNA polymerase that synthesizes new DNA. That is not synthesizing DNA, so that is also incorrect. Oops. Hold on. The answer, I don't know why that's going D. There we go. The answer is D. It's topoisomerase that relieves tension in the overwound DNA in front of a replication fork. All right. Moving on to number 12. Actually, number 12 is the same diagram that goes through 12 through 17. So I am going to actually, going to go ahead and stop recording.